let me talk about sequence alignments today. So, what is sequence alignment and what is its importance? So, um, so what is sequence? Sequence means this is a stretch of letters. In case of nucleotides, there are only A, T, G, and C, these 4 letters, how they are arranged inside the nucleotide. And in case of proteins, these are the 20 different alphabets are there. And the letters are generally the single letter codes. There can be a triple, 3-letter codes also, but that is not important for sequence alignment. Now, what is sequence alignment? Sequence alignment means it is a measure to check how 2 sequences are similar or dissimilar. Now, in case of biological or experimentations, sequence alignment plays a very vital role. Why? Because as per evolution, if 2 sequences, I'm mainly talking about the amino acid sequences of proteins, if 2 sequences have similar arrangements of letters, the 2 sequences might have the same structure and the 2 sequences might have the same function. So from the sequence alignment, we can identify how similar the 2 sequences are or how dissimilar the 2 sequences are. So that is one of the fundamental goals of biological sequence alignments. And now the sequence alignments can mainly be of 2 different types. One is called the local alignment and the other one is the global alignment. Local alignment means alignment of a portion of a sequence, and global alignment means alignment of the entire sequence. So I can show you diagrammatically what is what. So let me share my screen with all of you. The screen is visible, right? All of you can go to Google.
Yes, it's visible.
Yeah. Thank you. All of you can go to Google and just type local and global alignment. And you will get to see so many different types of alignments. Let me show you one of the alignments. Not going to the YouTube. Yeah. See, this is called a target sequence. This is called a query sequence. Query sequence, is it visible to all of you, right? All of you can see. Okay. So target means the sequence which is present in the database and with which you would like to align your sequence. And query sequence means it is a sequence of your choice. So if you look at here, this global alignment means alignment from the first letter to the last letter. See? And local alignment means alignment between only these portions, only a specific portions. And in case of sequence alignments, we have a complete match, exact matches. These are the exact matches, and these are the mismatches, the dots. Mismatches are represented by the dots. So, the matches and mismatches, matches means where 2 sequences are exactly identical, and mismatches means where the sequences are not identical, right? Now, the sequence, in case of sequence alignments, what is generally done is you take a query sequence, that means sequence of your choice, and you try to analyze the sequence. You try to align the sequence with the sequences that are present in databases. And the sequences that are present in databases, those sequences are referred to as target sequences. And one of the best tools for sequence alignment is the basic local alignment search tool, or the BLAST. I will give you a short brief demonstration of BLAST, which you can perform online. And in the next class, you will see the demonstration of BLAST in Unix platform. See. Now, this is the sequence alignment 2. This is the BLAST. You have several different options of BLAST. If you have only the nucleotide sequence, you have to use this one. It aligns a nucleotide with a nucleotide. And it is a protein BLAST. It aligns a protein with a protein. And this is a very important one, BLAST-X. What it does is, you have the nucleotide sequence, but you don't want to align the nucleotide. You want to know what type of proteins it codes. So it is called translated nucleotide. The nucleotide is translated computationally to generate the protein sequence. And then that protein sequence is used to use as a query. And it is the T-BLAST-N, it is the other way around. Like you have the protein sequence and the protein is back translated to the nucleotide. And then you do the nucleotide BLAST using the translated nucleotide as the query. Everybody got my point? Everyone? Any question you have? Yeah, in chat box, I think a couple of questions. Okay. Oh, repeat. Okay. Yeah, definitely. See. The BLAST means it is a sequence alignment tool. It is a tool with which you align two sequences. I will show you. If you have a nucleotide sequence, you should use this nucleotide BLAST, where your query is the nucleotide only. If you have the amino acid sequence of a protein, you have to use the protein BLAST. That means your query will be the amino acid sequence of the protein. Now say you have a different problem. You have the nucleotide sequence, but you don't want to align the nucleotide sequence, but you want to know what protein it codes. So in that case, what you do is you translate. There are mechanisms, there are algorithms to convert the nucleotide sequence to a protein. And with that mechanism, the nucleotide sequence is converted to a protein, and that protein sequence is subjected to BLAST. The protein sequence is then used as a query, and the protein BLAST is performed. And this one, it is the other way around. T-BLAST-N. So what is happening here is that you have the protein sequence, but you don't want the protein sequence. You don't want to align the protein sequence, but you want to know what nucleotide it is coming from. And you want to use that nucleotide information in the nucleotide BLAST. So it is the T-BLAST-N. It is the opposite of this one. So in this case, you have the protein sequence, you convert the protein sequence to nucleotides, and then that nucleotide information is used as an input, is used as a query to the nucleotide BLAST. Is it clear? Anything in the chat box? Let me see if anything in the chat box. Okay, let me see. 2 new messages. Okay. Okay. Now, see, in general, our main area of importance, or rather, our area of interest is the protein. Now, why protein? Because proteins are the main functional elements, or the functional parts, or the functional biomolecule. Nucleotides code for the protein, fine. Nucleotides only code for the protein. Nucleotides generate the protein, but proteins perform most of the functions, right? So in general, we perform the protein BLAST. So let me show you how you do the protein BLAST. See, here, you can type the sequence, or you can copy and paste the sequence. Let me show you. Okay, let me go back and let me go back to NCBI. And let's go back to protein. And then say, let us take a protein which is named as SOXR. This is an arbitrary protein. Okay. Let us take the faster sequence. Let us copy this faster sequence from here. Let us copy and paste it here. You just need to do a little bit of tweaking just to remove these positions to make it continuous. See, this is the faster sequence. And I think you remember what a faster sequence is. It starts with this greater than sign. And why you should use faster sequence? Because almost all of the tools, all of the biological tools or the bioinformatics tools, they need a sequence which is expressed in the form of faster. That means which starts with this greater than sign. And this is the name of the protein. And this is the accession number, and this is the organism from which it is coming. And these are the single letter codes of the amino acids. Now, see, this one is an important one. You need to consider this. This is called non-redundant. Non-redundant means all the databases that we have that are present. If you would like to search in a specific database, say at one of the databases that you were given demonstrations, that database is a UniProt. You can search that UniProt also, because if you click here and if you come here, you can press UniProt. So you can search this sequence against the sequences that are present in UniProt. So in this case, this is your query sequence and the target sequences are present in UniProt, but it is definitely your choice. You can use UniProt or you can use non-redundant. You can use anything. That is, it depends on the experiment that you want to conduct. And there are several options. We have the BLAST-P, and then there is this option, BLAST. The result will come, but it takes some time because it is searching against all these, and these are the results. And these are the 4 sequences that are present in UniProt. See, let me explain this one. These are the results. These are called the target. And your query sequence is the one that you copied and pasted in the query box. You need to remember this. This is code. This code is giving you an idea about how related the 2 sequences are. The higher the score, the better is the alignment. Then comes query coverage. Query coverage means how many amino acids are found to be in the alignment. I will show you, then you will understand it in a better way. E-value. E-value is an expectation value. E-value means it gives you an idea about how reliable the alignment is. The lower the E-value, the better is the alignment. And this is the percent identity. That means how many amino acids are exactly identical. And it is the length. It is the length. So this is called the graphic summary. See, if you can do a mouse over here, you will get to know the details. Details of these results. so on and so forth. See. And this is the most vital part. This is the sequence alignment. See, this is the query, and this is the number of amino acids. It starts with the first amino acid and ends, the first line ends in the 60th amino acid. The second line starts with the 61st amino acid, ends in 120th amino acid, and so on. And see, this is query, this is the subject or the target. This is the expectation value. E means 10. It is 3 into 10 to the power minus 109. This is the identity. See, there are 153 amino acids among the total 154 amino acids, which are exactly identical between the two. There is only a single amino acid which is different. So it is 99% identical. Similarly, positive. Positive means how many amino acids are related to them physicochemically. For example, see, this L and this L, these two are identical. But here, see, there is C in the query sequence and R in the subject. These two are not identical, right? These two are not identical. So that is why it is 153, not 154. And gap, I will come to that. See, here you will get to know what positives are. These are the positives because aspartic acid, this is D is aspartic acid, E is glutamic acid. These two have similar physicochemical characteristics. So they are called positive. Isoleucine and valine, they have similar physicochemical characteristics. So they are called positives. So up to this, there are no gaps. I'll show you what gaps are, but one important aspect is, okay, before I move further, any question? Any question from any one of you? Let me see in the chat. Okay, no question till now. Okay, one question I find. One message. One message. Repeat on positives. Okay, sure. See, positive. Positive means two amino acids which are nearly identical in terms of their physicochemical characteristics, like isoleucine and valine. Both of them belong to the category of amino acids, belong to the category of amino acids which are non-polar in nature. Just a second. Can you take this call just for a single moment? Hello? আমি একটু ক্লাসে আছি, আপনি আমাকে একটু পরে ফোন করুন 10 মিনিট। Yes, sorry about that. Okay, so and see here, K. K means this is lysine and R means this is arginine. Positive means a lysine and arginine. Both of them are basic amino acids. That means they perform the similar kinds of reactions. That is why they are positive, but they are not exactly identical. That is the problem. They are not exactly the identical. So there lies the difference between positive and identity. Let's see. Repeat on positive, yes. On which experiment we can go for non-reactive? Okay, I'm coming to your question, Krutika. But yes, I think it's Maganda. Maganda, your question. Did I answer your question properly? I'm sorry if I misspelled your name. Okay, and now, Krutika, on which experiment we can go for non-redundant databases? Okay, see, non-redundant databases, you can go for non-redundant databases when you have a completely unknown sequence, like you just have the sequence and no information about it. Then you should go for non-redundant databases. Sometimes it may so happen that through a sequencing experiment, you generate the sequence, but you have no idea about it. Okay, Krutika? Did you get your answer? Okay, fantastic. Okay, so let me again go back and do the same alignment with non-redundant databases. And generally, it is better to use the non-redundant database. It takes some time because it is searching so many databases, and non-redundant database means it is not a single database. It is searching in UniProt, it is searching in PDB and all other databases that are available. So definitely it takes time. Yes, we got the results now. See, the results will be a little bit different because see, there are 100. In the previous case, how many results did we get? We got only 4 results. Let us go directly to the alignments. These are the alignments. These are the alignments that we have. What I need to show, what I'd like to show is the alignment with gaps. None of these alignments have any gap. Alignments are showing up, they're coming. See, because we have so many alignments, more alignments are coming up. But all these alignments, see, none of them have any gaps. So let us do one thing. I need to teach you the gaps, right? So let us use this one, protein data bank, and perform the sequence alignment again. See, they are taking more time here because in protein data bank, there are not many similar sequences are available. Let's proceed in its own way, in its own pace. So let us take a couple of questions. Any questions? Any doubt? Any doubt you have? Okay, one question. It's taking time. Sir, what about the assignment submitted? Okay, the assignment that you submitted, they will be analyzed by the IBT team. Okay, let me show you. I think see here. You can see this is an alignment, and there are gaps here. Now, gap means in one of the sequences, you have the letters, and in the other, there are no letters. This is called gap. And gap plays a very vital role. Gaps are the mutations that are present in proteins or in nucleic acids. And gaps actually reflect how many mutations are there. And gaps represent the deleterious mutation. See, there are no nucleotides or no amino acids here. So this portion is entirely deleted from this sequence. Here also, this portion is entirely deleted. But in parallelly, this portion is added. This portion is added here. So this is called insertion deletion mutation. This is insertion, this is deletion. So it is also referred to as indel or insertion deletion mutation. Yes, global alignment. Global alignment, I think it is not coming yet. It's taking time. So let us go back. Yes, again, probably you get the result faster here. See, if you see here, it's taking time. It depends on how many. Yes, yeah, you got that. So this is a global alignment. See, it starts from 1 and ends in 154. So it covers the entire sequence. This is the global alignment. This is not partial. This is a global alignment. And in case of UniProt, you found local alignment because there were not 100% matches. See, in all these are the global alignments, the alignment of the entire proteins. See, okay, so you got your answer okay, yeah, sure. See, the protein has a total of 154 amino acids. So it starts with 1 and ends in 154. And see, the entire portion is aligned. Query with subject, query with target. So this is the global alignment. 2 unique messages. Yes, has, so should we say that global alignment? Yes, has, you can say that in global alignment, there are 99 to 100% matches. Definitely, you can say that. But it also varies. Like, if you have two very dissimilar proteins, in that case, you can perform global alignment. But in that case, the percentage identities may be 50% or 60% also. So in general, what I'm saying, see, in biology, you can never say that this is 100% correct, this is 100% wrong. There is always like 50-50 chances. In biology, 2 plus 2 is not always equal to 4. So in general, global alignments, there are 99 to 100% matches. But if you have two very dissimilar proteins, in that case, you can perform global alignment, but the exact matches will be around 50 to 60%. Here, the two proteins are identical, very similar. And any other question? So, yeah, on what basis we say that it is an indel? It can simply be a deletion insertion. How do you know which one is wild type? And that you should have some idea about. See, bioinformatics means you must have some idea. The target, you should know the target. Because you cannot compare an unknown with another unknown. That makes no sense. That is the basis. You cannot compare a hemoglobin with a SARS-CoV-2 protein, a spike protein of SARS-CoV-2. At least you have to have some amount of idea on to which protein you are going to align your unknown protein. Okay, any other question? Yeah, thank you. I think this is Kanvi. Thank you, Kanvi, for making this group. And you can type your questions here. So let us conclude here for today. And you will have your regular class, I think, from 4:30 p.m. according to Indian time. And you can type your question in the WhatsApp group. And in the next class, you will get a demonstration of this entire thing in the Unix platform. What I showed you is in the Windows platform. Unix platform, it will be a little bit different. But that is interesting also. You should know that. Yeah. Till then, thank you, everybody. Thank you for being with us. And I'm enjoying interacting with you all. Thank you.